Welcome to Chapter 12, which is going to cover DNA and RNA. First, we're going to talk about what is the role of DNA. One of the roles as DNA is to produce proteins. Role number 2 is to form the structural units of cells. And role number 3 is to control all chemical processes that go on within the cells. DNA contains genes, which are then in turn sequences of nucleotide bases. And we're going to examine a nucleotide once again here. You've already learned it in Chapter 2, but we will be reviewing it in Chapter 12. These genes code for polypeptides. And if you remember from Chapter 2 when we learned about nucleic acids, we mentioned that poly means many, and actually peptide is the bond that is going to attach things together. And in this case, in order to make a protein, we're going to bond amino acids. But that will come later in the notes. Proteins are used to build cells and do much of the work inside the cells. And this is something that you learned back in Chapter 2. Proteins are made of amino acids, and amino acids, once again, are linked together by peptide bonds. There are 20 different amino acids that are possibly going to link together to form proteins. And once again, a polypeptide are amino acid chains. So these are very long and complex. DNA is found inside the nucleus. And if you recall from your previous semester, the nucleus is the control center of the cell. It's in the center of the cell. Proteins, however, are made in the cytoplasm of the cells by organelles called ribosomes. And ribosomes were either free-floating within the cytoplasm or they were attached onto the endoplasmic reticulum. DNA's code must be copied and taken to the cytosol. You'll see the term cytosol, and this equals cytoplasm. So cytosol means the same thing as cytoplasm. In the cytoplasm, this code must be read so amino acids can be assembled to make polypeptides, or otherwise known as proteins. This entire process, from beginning to end, is called protein synthesis. So the entire information that you're going to be learning in Chapter 12 is going to talk about protein synthesis from beginning to end. What are two functions of DNA? Number 1, they store and use information to direct activities within the cell. And number 2, they copy themselves exactly for new cells that are created. So the new cells that are created are going to be identical to the original cell. Now we're just going to review something about the structure of DNA that you learned back in Chapter 2. DNA is a long molecule made up of units called nucleotides, which are repeating subunits and monomers. The DNA molecule consists of 3 parts. They are a 5-carbon sugar, and in DNA's case, it's deoxyribose, a phosphate group, and a nitrogen base. So if you recall from Chapter 2, the structure of a nucleotide has the phosphate group, the 5-carbon sugar, and then the nitrogenous base. There are 4 nitrogenous bases that make up the code for DNA. The phosphate group and the sugars will stay the same. The 5-carbon sugar deoxyribote and the phosphate group are the same in every nucleotide, but once again, the nitrogen base changes. And the nitrogen bases are as follows. There's adenine and thymine, and then guanine and cytosine. So those are the 4 nitrogen bases that make up DNA. What separates, or what's the difference between these 4 nitrogen bases? The single-ringed nitrogen bases are called pyrimidines. They are thymine and cytosine. So they just have 1 ring. The double ring are the purines, and these are adenine and guanine. And as you can see by their structure, thymine and cytosine have 1 ring. Adenine and guanine have 2. Purines can only pair with pyrimidines, and 3 hydrogen bonds are required to bond guanine and cytosine. So, guanine always bonds with cytosine, and they are held together by 3 hydrogen bonds. These bonds are very easily broken, so that when replication takes place, it's very easy to split them up. 2 hydrogen bonds are required to bond adenine and thymine. And once again, you can see there's 1, 2 hydrogen bonds. Thymine always bonds with adenine. So A always bonds to T, and C always bonds to G. Chargaff's rule, once again, states that adenine must pair with thymine, and guanine must always pair with cytosine. And the bases form a weak hydrogen bond. So once again, the weak hydrogen bond is going to attach this base to this base, and this base to this base. Here we have 3 between guanine and cytosine, and you've got 2 between adenine and thymine. DNA forms what are called anti-parallel strands. And if you look at the strands, they're going in the opposite direction. One strand of DNA goes from 5 to 3. And the other strand is opposite in the direction going 3 to 5. So here you have the backbone of DNA with your sugar and phosphates alternating on either side, and then you have your bases that go in the middle. Once again, guanine always bonds with cytosine, and adenine always bonds with thymine. And when we're looking at when it said 3 to 5, you'll notice the way that the sugars are oriented here. That's what they mean by 3 to 5. You see, they're going in the opposite direction. How did they find DNA? Where there was a woman named Rosalind Franklin who took diffraction X-ray photographs of DNA crystals. And this is a very famous photograph called Photograph 51, which showed an X-shaped pattern of twisted strands that are known as a helix. And there would be the picture of the photograph, famous photograph 51, that shows that X down the center. In the 1950s, Watson and Crick built the first model of DNA using Franklin's X-rays. They built a three-dimensional model using cardboard and wire to explain the properties of DNA. Watson and Crick made this discovery in 1953. Watson and Crick went on to eventually win the Nobel Peace Prize, and because Rosalind Franklin had died due to cancer from all of the radiation exposure, she was never given credit towards the discovery of DNA. So, DNA is two strands coiled together, and it's called a double helix. It is not a twisted ladder. If you use the term twisted ladder on any of your worksheets or any of your tests, I will mark that answer wrong. We need to use the scientific name double helix. Once again, the sides are made of a pentose sugar, deoxyribose, and they're bonded to phosphate groups. The center, made of the nitrogen bases, is bonded together by weak hydrogen bonds. So sometimes DNA is represented by this coil-looking or spring-looking diagram, and sometimes it looks like a ladder. The legs of the ladder or the sides of the ladder are the sugar and phosphate groups, and then the rungs of the ladder are sometimes considered the nitrogenous bases, which is A, T, C, and G. If we're looking at the real or the actual DNA molecule, it has a right-handed twist with each full turn consisting of 10 base pairs. So as we're looking on this, a full twist goes from here down to here. That is one full twist. So here there's half, and there is the other half. And then if we were to go to the other side, here's one half going down. So in between here, there are going to be 10 base pairs. So once again, one strand of DNA, the backbone of the molecule is alternating phosphate and ribose sugars, and then the teeth are the nitrogenous bases. A two-stranded DNA, the strands are bonded together to form a molecule of DNA, which then twist to form the double helix. And what we said before was hydrogen bonds are what attaches these bases together. And you have to always remember that guanine and cytosine always pair together, and adenine and thymine always pair together. And as you're doing this entire unit, they will remain color-coded in everything that you do to help you hopefully remember each of the different parts and pieces. And this concludes the structure of DNA.